Bismillahirrahmanirrahim. Assalamu alaikum, dear student. Welcome back to your channel, The Anatomy Canvas. Today we are discussing the histology of the male reproductive system, part 2. And in this class, we will discuss the excurrent duct system. So objectives of the today's slide are to discuss the efferent ductules, histology of epididymis, and histology of the vas deferens. So first, we will discuss little bit revision from this class, from the previous class. This is the structure of the testes covered by the tunica vaginalis, and under tunica vaginalis is the tunica albuginea, and then the dense tunica vasculosa present, which divides the substance of the testes into different lobes. And then in the lobular structures of the testes, the seminiferous tubules are present. These seminiferous tubules then connect with through the reedy testes to the efferent ductules. So reedy testes are receive fully formed spermatozoa, which leave the seminiferous tubules by the straight tubules and enter the interconnected channels of the reedy testes. This is the histological diagram. These are the histological sections of the seminiferous tubules, and this portion indicates the reedy testes. In this diagram, you can appreciate that the anastomosing channels which are present in this area are the area of the reedy testes. These anastomosing network are lined by the low cuboidal epithelial cells and surrounding connective tissue, which is formed by the mediastinum testis. It is dense and it is formed from the tunica albuginea. Again, another diagram showing you the different features. The outer is the tunica vaginalis, then the tunica albuginea, the tunica vasculosa. These are the testicular lobules formed by the seminiferous tubules. These tunica vasculosa forming the septa. These seminiferous tubules connect via straight tubule erecti to the reedy testes, and then the sperms enter through reedy testes, passing through reedy testes into the ductuli efferent. So ductuli efferents in the head region, they are lined by the pseudostratified columnar epithelium with cilia. So as the cells sperms over here are non-motile, so this ciliated epithelium help these cells to move from the area of the efferent ductules and enter into the head of the epididymis. The lining epithelium in this area is pseudostratified columnar, and it is covered by the smooth muscle cell layer. Another diagram showing you the histological section of the efferent ductule. This is the pseudostratified epithelium of the efferent ductule. It showed typical saw-toothed or serrated appearance, lined by both the columnar cells and the cuboidal cells, surrounding a small amount of the connective tissue, and then the circularly arranged smooth muscle fibers are present, and in between the smooth muscle fibers, the smooth muscle cells, elastic fibers are also present. Efferent ductules, another same diagram. The reedy testes lead into efferent ductule that lead into the head of the epididymis. So when these efferent ductule enter into the head of the epididymis, they show convolutions and form the coni vasculosi, and through which they enter the base of the head of epididymis. They have the scalloped or the serrated appearance over here, the cells having cilia which beat to help move mature sperm to the epididymis. So next in the excurrent duct system is epididymis, and this epididymis has head, body and tail. It is a highly coiled one tubule nearly 4 to 6 meter long, and it is lined by the pseudostratified columnar epithelium with stereocilia. These stereocilia are unusually long microvilli. They do not have a structure typical exoneme of the cilia. The principal cells and the basal cells, two basically types of the cells are present in epithelium of the ductus epididymis. The principal cells having stereocilia and basal cells. Occasional intraepithelial lymphocytes may be present, and these lymphocytes are called the halo cells. Two layers of the smooth muscle cells are present in epididymis, and these are the thick muscle core. Function of the epididymis is to for the maturation, the storage and the transport of spermatozoa. So these are the histological diagram. This area shows the section of the epididymis, and this area shows the section of the efferent ductule. So in this diagram you can appreciate this is the higher magnification of the epididymis. So you can appreciate the pseudostratified epithelium with stereocilia. And another very important identification point is that the inner profile of the ductus epididymis is very well defined. In all these sections you can appreciate that they have very well defined, very clear inner profile as compared to the efferent ductules or the seminiferous tubules. So efferent ductules present over here are lined by the pseudostratified epithelium with the cilia. Another histological section showing you the ductus epididymis, and the ductus epididymis lined by the pseudostratified epithelium with the stereocilia. So surrounding connective tissue is present, and this is the low magnification and this is the higher magnification, and this area shows the low magnification of efferent ductules and this is the higher magnification of the efferent ductule. So you can appreciate the pseudostratified epithelium, sometimes occasional halo cells which are intraepithelial lymphocytes are also present over here. Another histological diagram through the testes, you can appreciate the different seminiferous tubules lined by specialized type of the stratified epithelium, and you can appreciate that inner profile in case of the seminiferous tubule is not very well defined as compared to that of the epididymis. Also in the lumen of the seminiferous tubule you can appreciate the bunch of the spermatozoa are also present. This is another diagram, the epididymis head, body and tail of epididymis, now continuing as the ductus deferens. So now we will study the ductus deferens. Ductus deferens passing through the inguinal canal and inguinal ring enters into the spermatic cord. So in spermatic cord, ductus deferens is present along with the vessels, the muscles, nerves and the lymphatic vessels, etc. So ductus deferens histologically it is lined by the pseudostratified columnar epithelium. Underneath epithelium there is a laminae propria and there are very well defined thick muscle coat which is organized into three layers: inner and outer longitudinally arranged smooth muscle layer and middle circularly arranged smooth muscle layer, and outer to this muscle layer is the adventitia which is loose connective tissue. This is the histological diagram of the ductus deferens. In this diagram, you can appreciate the lumen of the ductus deferens, the mucosa, which is formed by the epithelium and the smaller amount of the connective tissue. Epithelium is a pseudostratified as in other parts of the excurrent duct system, and then the longitudinal muscle coat, the circular muscle coat, and another longitudinal outer muscle coat. So this thick muscle coat is the identification point for you under the microscope, and outer to this thick muscle coat you can appreciate this is the adventitia, which is loose connective tissue having blood vessels, having adipose tissues, etc. In the inset you can see at low magnification this is the spermatic cord, in which you can see the vas deferens, large number of the blood vessels and nerves are present which are forming the pampiniform plexus over here. Another histological diagram this is again the histological diagram of the spermatic cord, in which you can appreciate the ductus deferens is present. This is the lumen of the ductus deferens formed by the mucosa, then the thick muscle coat and the adventitia, and surrounding this adventitia there are the large number of the blood vessels which are present along with nerves and lymphatics forming the pampiniform plexus in the spermatic cord. This is the high magnification of the same slide. You can appreciate the inner profile, that is a pseudostratified epithelium having the bunch of spermatozoa in the lumen of the ductus deferens, and still at higher magnification you can see pseudostratified epithelium, inner longitudinal muscle coat and middle circular. Here inner longitudinal muscle coat, middle circular and outer longitudinal muscle coat as well. So Another diagram of the ductus deferens present in the spermatic cord. You can appreciate the lumen, and because of the thick muscle coat which contract lumen sometimes thrown into folds. So you can appreciate the folding of the lumen in this slide as well. And lumen it is formed lined by the mucosa, then the muscularis, and then the adventitia, and large number of the blood vessels which are present in the spermatic cord. Another diagram of the ductus deferens, its mucosa formed by epithelium, connective tissue, the inner longitudinal, the middle circular, and outer longitudinal muscle coat, and then the adventitia. Another diagram at still further higher magnification, you can appreciate its epithelium. You can appreciate smaller amount of loose connective tissue having large number of the nuclei present over here, and then the inner longitudinal muscle coat. So as the vas deferens enters, it joins the duct of the seminal vesicle and form the ejaculatory duct and ampulla. So ejaculatory duct is lined by the pseudo-stratified columnar epithelium having the lamina propria. Only the muscular layer are present in the ampulla. The longitudinal muscle coat disappear near the origin of the ejaculatory duct. So here the excurrent duct system is completed. These are the references, that is Histology, a text and atlas by Michael H. Ross. And till the next class, Allah Hafiz, and thank you very much for your attention.